So welcome back. So today we want to talk about the renal system, and this is going to be the third of our lectures about the renal system. And the things that we want to discuss today is how the kidney then uses the tubule transporters to reabsorb materials, that is to take it from the filtrate back into the body or into the blood, and also to secrete materials bypassing the filtration route that we talked about the last two times. So the specific learning objectives are first that we're going to explain the importance of the peritubular capillary in the kidney cortex. And secondly, we want to describe the cellular mechanisms for transport of materials from the tubule, the interior of the tubule, that is within the lumen, across the cells and into the blood. And we're going to use the glucose transport as our example. And then very briefly, we'll talk about moving bicarbonate across this system. Third, we want to talk about the transcellular transport, that is the movement across cells versus movement between cells, the paracellular pathways. And this is the way that water then will move across this particular region of the kidney. And we'll also talk very briefly about the solvent drag of potassium. Fourth, we will define the maximum transport rate and the transport threshold for items that are using transporters. And five, we'll consider secretion. And secretion only very, very briefly, and then also we'll talk about secretion, and particularly of secretion of potassium, and again, very briefly. So let's revisit then our tubule, and that's what's drawn here. So we have the afferent arteriole, which is feeding into the glomerulus, and that's our first capillary bed. And that first capillary bed is drained by a second arteriole, and that's the efferent arteriole. And the blood then drains into the second capillary bed, which is called the peritubular capillary. And the peritubular capillary runs along the renal tubule within the kidney. We also have the renal tubule, and that's what's diagrammed here. And around, circling around the glomerulus, we have Bowman's capsule, and that's this. And then from Bowman's capsule, we enter into the rest of the renal tubule, and this is the first region is the proximal convoluted tubule, and then we'll go through the loops of Henle and eventually lead to the distal tubules and the collecting duct, and then out through the ureter. So the processes that we're talking about is at the beginning, between the glomerulus and Bowman's capsule, we have this filtration, and the filtration will move solutes and water into the lumen of the renal tubule. And we know that that's going to be governed by specific pressures, and that's going to be hydrostatic pressure minus the oncotic pressures. So now we want to move materials from the renal tubule back into the blood. And this is going to be very important because within 24 minutes, we can completely exhaust the cardiovascular system if we do not bring the fluids, the water, and particularly back into blood. We also need to be balancing ions, and that is and particularly with bicarbonate, we are filtering bicarbonate freely across the glomerulus, and we need to move that bicarbonate back into the blood to use it as for pH buffer. And so reabsorption then, which is what we'll talk about predominantly today, is how we're moving solutes from the lumen of the tubule back across the cells back into the blood. And then there's also a way that we can bypass filtration and move materials directly from the blood, from that peritubular capillary, directly into the renal tubule, and that's called secretion. So what are the things that are governing this particular region? So most of the movement then, the reabsorption, filtration and reabsorption is going to be governed by pressures. And that's what's diagrammed here. So you recall we had this portal system where we have the afferent arteriole, which is on one side of the glomerulus, the first capillary bed, and then we have the efferent arteriole, which is on the second side of the capillary bed. And that's different from what we see in normal places within the body, where we have an arteriole feeding into a capillary, which is then feeding into a venule. Within the nephron, then, this afferent arteriole and the efferent arteriole, their diameter of the lumens of those vessels are regulated independently. And we can maintain the hydrostatic pressures within the glomerulus such that it always exceeds the oncotic pressure. And when hydrostatic pressure is higher than the oncotic pressure, and it's higher also to Bowman's capsule, the resisting pressure of moving fluid into Bowman's capsule, then we have filtration. And across this region of the nephron, there's always, always, always filtration. When we move into the peritubular capillary, now we have a situation which is more analogous to what we see in the rest of the body, and that is that after the efferent arteriole, the hydrostatic pressure is now lower than the oncotic pressure. This means that the oncotic pressure is attracting water back into the peritubular capillary, and that occurs all the way along the peritubular capillary, and so that occurs all the way along the renal tubules. And this is then reabsorption. We're removing fluids then back into the blood all the way along this, the renal tubule. And this is particularly important within the proximal convoluted tubule. So if we consider the proximal convoluted tubule, and that's what's drawn here, I've drawn two of the cells, the epithelial cells of this region, where the lumen is here and the blood is here. The blood space is here. And there is a very small interstitial space between the cells, the basal section of the cells, the basal surface, and the blood capillary. And this, of course, is the blood capillary is our peritubular capillary. The cells that line this tubule region in the proximal convoluted tubule are asymmetric in the expression of transporters, such that transporters which are located on the luminal surfaces of these cells are different from the transporters which are located on the basal surfaces of the cells, or the surface that's facing the blood. And what I've diagrammed here is one such transporter, and this is the glucose-sodium transporter. So this is a co-transporter, and it's a symporter. It's moving glucose and sodium in the same direction. As the sodium is moving into the cells, the glucose is piggybacking onto this movement, and then the glucose, once it's in the cell, can exit at the basal surface of the cells and enter into the blood, simply going down its diffusion gradient. The sodium is also moving down its diffusion gradient due to the action of the sodium-potassium ATPase, which is located on the basal surfaces of these cells, where we're extruding sodium in an active manner. We're using ATP to move 3 sodiums out of the cell for 2 potassiums to enter the cell. When we have a system where we have a cotransporter, which is linked to an active transport of one of the solutes that the cotransporter is using, then this is called secondary active transport. And if you recall, we discussed these in the second lectures of this course. Now, as we're moving the sodium and the glucose across these cells, we're generating an osmotic gradient, and so water will then move down its osmotic gradient across the cells. We have aquaporin channels present within the luminal surfaces of the cells. So water then will follow the sodium to enter into the blood. And the movement of these ions, of the sodium and the glucose, will be isoosmotic as it's moving from the lumen to the blood. The same thing will happen for a movement of amino acids and for other small solutes going across this particular region. Now, one of the other important items that has to be moved from the luminal surface, from the lumen, from the filtrate, is bicarbonate. So bicarbonate is freely filtered, as are protons, and they will enter into the tubule lumen. And in the tubule lumen, on the surfaces of these cells, the proximal convoluted tubule epithelial cells, we have carbonic anhydrase activity. Carbonic anhydrase, as you remember, will generate water and CO2 from a proton and bicarbonate. And the proton and the bicarbonate cannot move into the cells, but the water and the CO2 can freely enter into the cells. Once they're inside, then we will regenerate the proton and the bicarbonate because there's carbonic anhydrase activity within the cells. The bicarbonate is removed from these cells using the antiporter, which is the bicarbonate chloride antiporter, which is located on the basal region of these cells, and bicarbonate then enters into the blood. The proton is going to be extruded from the cells back into the lumen of the tubule, and this will occur in exchange for sodium. So this is an antiporter the proton is leaving back into the tubule, and sodium enters the cells. Now, the rate of transport of materials across this region is saturable because we are using transporters, and that's what's shown here. So with increase in the solute concentration, which is along our x-axis, as that increases, then the transport rate, which is shown here on the y-axis, is increasing linearly. But eventually, we saturate all of the transporters, and at that point, we then have a maximal saturation of the transporters, and so we have maximal rate of transport of the material. When this occurs, and we have all of the transporters occupied, we are then said to be at threshold for that particular substrate. So, for instance, if we're talking about glucose, as you all know, glucose is freely filtered. It enters into the filtrate, and all of the glucose will be reabsorbed across the proximal convoluted tubule, and none will be found in the urine. But in the cases of an individual who has diabetes mellitus, they have very high levels of circulating plasma glucose. And this plasma glucose, when it's filtered and enters into the filtrate, then binds to the transporter for the sodium glucose transporters and will actually saturate all of the transporters. So any of the glucose which is left over, which cannot bind to the transporters, stays within the renal tubule, and it is then delivered to the later regions of the tubule, which do not have transporters for sodium and glucose. That means that the glucose and the glucose then remains within the lumen of the tubule. It holds water because it's osmotically active, and these individuals then are going to have a problem. They're not going to be able to concentrate the urine. So they will urinate frequently, and they'll be constantly thirsty. So some of the terms that you need to keep in mind then is, one, the transcellular movement. And transcellular movement is moving a material across the cells, going from the lumen across the cells and into the blood. This is usually done by secondary active transport within the proximal convoluted tubule. And this is the region where almost all of the materials that are within the filtrate are reabsorbed and moved back into the blood. That means all of the glucose, all of the amino acids, even very small proteins are broken down to amino acids and moved across in this region. And we have the movement of chloride and urea. But chloride and urea will move by facilitated diffusion. Because as we're moving ions and glucose and so forth, these are osmotically active particles. They move across the cells, water follows them, and then the chloride and the urea become more concentrated within the filtrate. That generates then a diffusion gradient for them, so they now are higher within the lumen. Their concentrations are higher in the lumen, and they can then move across the cells to enter into the blood. The second type of movement we've never discussed, and this is called paracellular movement. Paracellular means that we're going between the cells. So if I draw two of these cells, the cells are connected on the luminal surfaces by what are called tight junctions. The tight junctions are little seals that prevent the luminal content from leaking across between the cells to the blood, and the blood surface is over here. This is extremely important in the gut, where we have one cell connected to the other, and all of the uptake of material from the gut has to occur across the cells to get into the blood. In the kidney, however, these tight junctions are a little leaky, and they're leaky to water. So water can move across the tight junctions in a paracellular manner, that is, between the cells. As water is moving between the cells, it is also setting up a concentration gradient for potassium. We do not have transporters for potassium located within the proximal convoluted tubule. And here, as the potassium concentration rises, it now generates a gradient, a concentration gradient for potassium to diffuse across this region. But potassium diffuses between the cells, so it's going in a paracellular manner. But the potassium is an ion that is very important to the body, and it actually has its own special name, and we call that solvent drag. But the solvent drag is the paracellular movement of potassium, and it's the movement of potassium between the cells, and it's moving because as we move water across these cells, we concentrate the potassium within the renal tubule. So let's switch gears slightly and talk about secretion. Secretion, as you recall, is the movement of materials from the blood space across the renal tubular epithelium and into the filtrate itself. So the secretion that I want to talk about is in the proximal convoluted tubule. So that's the same region we were talking about just a few minutes ago, where the majority of reabsorption of ions, water, so forth, is occurring in the proximal convoluted tubule. With the secretion, we're going to be moving materials that is directly from the peritubular capillary across the cells and into the lumen of the tubule. And the materials that we're moving are going to be organic compounds. These are going to use transporters which are sort of generic transporters, and they're located on the basal surfaces of the cells. So the generic transporter then is allowing this material to go from blood into the lumen of the tubule. This, again, we're going to be using secondary active transport as our means for moving this material. And the transporters, which are located here on the basal surface of the cells, are absent from the luminal surfaces of the cells. So the transport is unidirectional, and again, transport is going to be saturable because we have a finite number of transporters located within this region. So what are some of the materials that we move through the secretion? And one of them is that you move epinephrine. You also move norepinephrine across this region. Vitamins are taken across and cleared from the blood by secretion. Vitamins such as vitamin A, vitamin D, and so forth. We also have an instance where we're moving drugs, and drugs such as morphine and penicillin are secreted. So they are moving directly from the peritubular capillary across the proximal convoluted tubule epithelial cells and into the lumen. And then they will then be removed from the body in the urine. Now there's the special case of secretion that I wanted to talk about just briefly is that of potassium. So in the collecting duct, we have a cell which is called a principal cell. This principal cell is also located in the later region of the distal convoluted tubule. The principal cell reabsorbs sodium, that is, it's moving sodium from the lumen, from the filtrate, then across these cells and into the blood space. And in exchange, we're going to be moving potassium from the blood across the cells and into the lumen. So you notice that there's no charge gradient that's going to be established. We're just simply moving a positive ion, sodium, from one side to the opposite, and then moving potassium, which is a positively charged ion, in the opposite direction. Now, this entire region is using a sodium channel, which is moving the sodium, which is allowing the sodium to enter the cells, moving down its concentration gradient, which is established, of course, by our sodium-potassium ATPase. The potassium is moving in the opposite direction, is entering into the filtrate, and it is doing so also down its concentration gradient. As the sodium is extruded by the sodium-potassium ATPase, we're moving two potassiums into the cell. So we're increasing the gradient then for potassium in the inside of the cell, and we can then have the potassium diffuse through this channel out into the lumen. So if we deliver a lot of sodium to this region, a lot of sodium into this collecting duct, then we will drive this movement of sodium into the cells and across the cells and into the blood. And that, of course, will drive the sodium-potassium ATPase. And by doing so, we then increase the intracellular gradient for potassium. Potassium then will exit at a faster rate from the cells. So it's a way then of removing potassium from the blood in this particular region. Now, this particular region can also increase the removal of potassium by simply increasing the filtration flow through this area. And that is that as the sodium is being delivered, the sodium delivery is not as important as the potassium. We wash away the potassium, and now the gradient for potassium to exit the cells is increased, and potassium then will leave the cells at a faster rate. This will then drive the sodium-potassium ATPase, and we will then lose potassium into the blood. So we have two ways then of increasing the secretion of potassium. One is to have a delivery of high amounts of sodium to this region. And secondly is to increase the filtrate flow and delivering a high filtrate flow to the collecting duct. Now, the hormone aldosterone, as you all recall, is secreted by the adrenal glands, and this hormone regulates potassium levels within the body. This aldosterone is increased, its secretion is increased when potassium rises within the blood. And what aldosterone is doing is that it's acting on these principal cells within the kidney. It's increasing the copy number of the sodium channel, of the potassium channel, and of the sodium-potassium ATPase. So it's driving this entire system. And that's the way that aldosterone then is able to remove potassium from the blood. So what are our general concepts? So the first is that reabsorption moves the filtered solutes from the renal tubule back to the blood. The second is that this reabsorption of solutes occurs predominantly within the proximal convoluted tubule. That's our major region for uptake of water, for sodium, all of glucose, all of the amino acids, and so forth. Third, most of the solutes will cross this epithelium in a transcellular manner, and it'll be so by secondary active transport. Four, we have reabsorption within the proximal convoluted tubule occurs isoosmotically, and that the water crosses the epithelium via the aquaporin channels, which are inserted within the luminal region of the cells. And water can also cross in a paracellular manner, and that is between the cells across those leaky tight junctions. And the potassium will move by solvent drag in a paracellular manner. So it's following the water in a paracellular manner. And that is, as water is moving, it's changing the concentration of the potassium in the tubule. And the potassium now has a higher concentration in the tubule. It can then diffuse across this region and into the blood. 5, secretion is the movement of solutes from the blood to the lumen, and this again occurs in the proximal convoluted tubule, and again predominantly by secondary active transport. And these are our organic molecules, our drugs, the vitamins, and so forth. And then 6, the secretion of potassium occurs in the collecting duct and also in the distal regions of the distal convoluted tubule. And this is in response either to an increased sodium delivery to this region or to a high filtrate flow. And that, importantly, aldosterone acts on these cells, the principal cells within this region, to increase the channels for potassium, the channel for sodium, and also the sodium-potassium ATPase. And so it drives then the movement of potassium from the blood and into the lumen of the tubule, and then eventually to be excreted by the urine, in the urine. Okay, so the next time we come in here, then, we're going to deal with the movements of water across the system. Okay, so see you then.